Like all right,
Okay. Well, let me know if you think this is at the right.
All right. Good evening, everyone. Can everyone hear me on well, it's for the Zoom. Can everyone hear me on Zoom? All right. Good evening, everyone. Thank you for joining us tonight. Okay. Oh, hi, everyone online. Thank you for joining. So happy fall, y'all. Sorry. Okay, I did write that down, I promise. But so I just want to get started with some announcements. Dr. McCarter, would you like to share any reminders or anything about our our meeting?
Just a reminder, First Coast Symposium, February 23rd and 24th at the Hyatt Downtown at 2023. And I promise, I know I promised last week, but I promise Noel is this close, just like less than an inch, this close.
She's saying this.
To having the website ready to go for registration.
Wonderful. Thank you. So I don't know if anybody recalls from the last time, but I requested some membership highlights. I think my email may be broken. But I did want to share a membership highlight just to give an example. She said it was okay with a grimace on her face. So I am going to highlight Kimberly McKean, who is our molecular supervisor at Baptist Health. And she's been busy, as I'm sure everyone is in their laboratories. And we just went live with our CT&G on our Panther. And she headed those validations. And we validated our new Genius reader, which was super exciting because it was about to die today, as well as a vaginosis panel. So she's been busy. And what I'd like to share about membership highlights is too, just background. I didn't go in, didn't ask her too much detail as to how long she's been around. But if you want to share that, that would be wonderful. And anything else, really just to highlight the membership, new team members that are learning new things, anything. That's what we're looking for. And so Kimberly's laughing over here. Nobody online can see her, but everyone here can see her. She's right there. Everybody probably knows her. Thank you for letting me pick on you. So please send me your membership highlights. Okay. Any announcements from the membership to share with the group? Anything exciting going on around here? Okay. Am I shouting? Oh, okay. Anything? No. Okay. So Matthew, would you mind sharing a current membership report?
Yeah. So the CUs have been entered for last month. So if you got any, let me know. All the membership checks, except for the ones that I received today, have been positive. So, but that was today. So that'll be going through, and we're probably after all this stuff that I'm receiving, maybe 55, 60 members. We're getting up there. Thank you all.
All right. Thank you. It's never too late to join Jam. And Beth, anything? Where's my Beth? Where is she? Over there. Okay. Thank you, Beth. Thank you, Lisa. Yes. So Beth was sharing about the RSVPs. I'm not sure if everyone can hear when we're talking out in the room. But even if you don't RSVP, don't feel like that doesn't mean that you can't come to the meeting, because you sure can. Just don't steal other people's food, please. Okay. So any last minute announcements? If not, I will pass it on to our sponsor. Lisa Ross from Shianogi Incorporated has graciously accepted sponsorship to this meeting. And so a big thanks goes out to you, and she'd like to say a few words.
All right. Hey, everybody. Hey, all virtual people out there. I see so many familiar faces. I'm so very happy to be back. I shared with a lot of people that I left Merck and went into a different pharmaceutical sales space, selling to anesthesia and surgeons. And I've spoken here before about how much I love this group in particular. And I had no idea how much I missed it. It was just wonderful to come back. But I'm back with Shianogi. My new product is so fun. If you don't mind, I'm just going to go to a quick little verbal explanation of it because a lot of people don't know it. It's called Petrosia. It's a gram-negative antibiotic, a cephalosporin. And it's a really fun drug. It all has to do with iron, hence the Fe in Petrosia. So what I didn't even know after years of antibiotic sales experience is that bacteria have to have iron to proliferate. I did not know that. Most of you probably did. So when they enter the body, they get iron from the bloodstream, just free iron that's floating around. And as soon as the body's immune response realizes that free iron is being depleted, it starts sequestering the free iron using something called lactoferrin. I'm sure everybody knows what that is. And then when the bacteria realize that there's not enough free iron, which they must have to proliferate, they did this really cool thing where they send out these little things called siderophores. I think of them like Pac-Man. And they attract iron. And when they encounter a bacterial cell, again, because they're outside of the cell, they're just like scavengers, there's something called the iron transport system. And the bacterial cell recognizes that as a nutrient and pulls it into the cell through the iron transport system. So the other way that things get in and out of cells is through porin channels. So people remember porin channel changes is one of the major mechanisms of antibiotic resistance. And so normally when a bacterial cell senses that antibiotics are entering it, it simply closes or deletes those porin channels, and beta-lactam antibiotics cannot enter the cell. So what this company did that is so clever is they attached one of these siderophores to a cephalosporin. So the cephalosporin can get in the normal way through porin channels. That's called passive diffusion. It just kind of soaks in through those openings. But when the cell closes that entry point, when it has an iron molecule attached to the antibiotic, it thinks, oh, look, a siderophore, and it pulls it into the cell, tricking the cell. So that's the quick and dirty on how this drug works. The name is brilliant. It's Fe for iron and Troja for, it's so cool. All right. So the coolest thing about it is also sort of its Achilles heel, testing. Because if you're using broth dilution media and a lot of the automated systems, there's problem with you have to use iron depleted media. So there was a problem with testing for a long time. And, you know, with a new antibiotic, testing is always the Achilles heel of every antibiotic. But they finally have disc diffusion testing is FDA approved. Nothing has to be iron depleted with it. And we've had Megan at Ascension just finished verifying it about a week ago. It was not that painful. Yeah. And Shianogi entered this agreement with LSI where you can get the panel for free. They provide support. And then Shianogi is just a fantastic company. I love it here. They are the first and only company I've ever heard of that has a medical science liaison who's solely dedicated to field microbiology. So if anyone wants to get really down into the weeds with Petrosia, I've got the guy for you. And then I have a new MSL on the way, as I was discussing with you. So that's it. If anybody wants information about getting that panel, like I said, it was pretty painless. They were trying to make it so verification to me in a microbiologist world is like taxes. Like, you have to do your taxes. Everybody knows how to do their taxes. Nobody wants to do their taxes. So they're trying to make it a lot easier with the support from the MSL, the support from LSI, the free panel. The only thing you really have to buy and pay for are the discs, which you'll be using in the first place. So that's it. And then I think Dr. Rowlinson is going to come talk to us about antimicrobial resistance. Right? She's next.
Thank you, Lisa. I appreciate it. Yes. So very exciting. It's always nice when we get help for validations in the lab. Okay, so I am introducing a fan favorite, Dr. Marie Claire Rowlinson. She rejoined the Florida Bureau of Public Health Laboratories as Bureau Chief in June of 2023, overseeing the 3 laboratories in Jacksonville, Miami, and Tampa that comprise the State Public Health Laboratory. Dr. Rowlinson provides administrative and technical oversight for diagnostic and surveillance testing at the Bureau of Infectious Diseases, newborn screening, and other activities that address public health threats. Prior to this, Dr. Rowlinson was the chief of bacterial diseases at the New York State of Public Health Laboratory, Wadsworth Center for 2 years. She was gone. And before that was the assistant director and CLIA laboratory director of the Bureau of Public Health Laboratories in Jacksonville for nearly 10 years. And I told her she really shorted us with this bio she sent me, but she's in tons of committees, tons of everything. She's awesome and amazing. And she's going to present on detecting and preventing infections with antimicrobial resistant organisms.
I think I can use this.
Yeah.
Just let me know that you can hear me.
Yeah.
And hopefully everyone online can hear me. I may walk around a little bit. So I may not be on camera the whole time. So I apologize for those that are joining virtually. Okay. Should I say it aloud to this? Signing in on another device. That's okay. Okay, perfect. All right, well, it's so nice to be back. Francis was like, was it really 2 years? And I was like, well, did it seem longer or did it seem shorter? And she was like, couldn't believe it had actually been 2 years. But I have no concept of time since COVID. I'm like, oh yeah, that happened 6 years ago. And then someone will say, no, that was in 2013. I'm like, oh, okay. Or I'll say, oh, I think it happened about 5 or 6 years ago. And they'll say, no, it was like 6 months ago. I don't know what happened to my concept of time. But anyway, I was in Jacksonville for nearly 10 years, went to upstate New York for 2 years, missed you all, missed the weather. Not gonna lie, didn't appreciate winter in upstate New York. And so it's really good to be back and be in this exciting new role, overseeing all of the labs. So I'm sort of less involved in a way than what I was in Jacksonville before, although I'm still their CLIA lab director. So anyway, so today what I wanted to talk to you about was antimicrobial resistance and healthcare associated infections. And I've got some data that I'm going to present from both New York, and I'll explain why in the process of my presentation, and Florida. So I wanted to start today with a case. So these were the headlines. CDC investigating deadly outbreak of multi-drug resistant Pseudomonas tied to eye drops. Drug-resistant bacteria tied to eye drops can spread person to person. Multi-state outbreak of antimicrobial resistant Pseudomonas aeruginosa. So what happened with this outbreak of carbapenem-resistant Pseudomonas aeruginosa? Well, in August of 2022, the New York State Public Health Lab, Wadsworth Center, identified a rare carbapenemase gene in a patient from a Connecticut long-term care facility. And in the process of doing testing, they did whole genome sequencing on this isolate and identified 2 resistance genes, one VIM80, which is a carbapenemase, and the other was GES9, which is an extended spectrum beta-lactamase. And neither of these variations of these genes had actually been seen in the United States before. So they were like, that's interesting. We should tell CDC, hey, we just got this CRPA and it has VIM80, GES9. It was reported to CDC. And then not much else happened for a couple of weeks. But a couple of weeks passed, and then CDC was getting notified about other clusters of CRPA. There was a cluster in Los Angeles, at LA County, and in the state of Utah. And these notifications were coming through health departments in those jurisdictions. And essentially what whole genome sequencing identified was that all of the isolates from these cases in the Connecticut long-term care facility, the Utah long-term care facility, and an eye clinic in LA County, was they were all CRPA, they were all sequence type 1203, and they all had these novel carbapenemase genes, the VIM80 and the GES9. And whole genome sequencing was, as well as saying what the sequence type was and showing what these genes were, was also able to show that isolates from these clusters were pretty closely related. So you see on the right-hand side, there's a timeline, and you know, things like this don't kind of happen over time. So actually, the specimen on that patient that was initially identified as having this VIM80 GES CRPA, I think that specimen was collected in May, but it's August by the time the whole genome sequencing happened and all that stuff. So between June and August of 2022, 3 facilities reported CRPA clusters. But at that time, they still didn't know, were they all the same thing? But by October of 2022, that's when these clusters were linked to one another. The whole genome sequencing had said these cases are closely related to one another. And CDC sent out what's called an EpiX, which is usually an email. It's the CDC's Epidemic Information Exchange. And it's a secure network for exchanging information with pertinent people. So health departments and so forth, labs, and HAI, AR programs, for example, in health departments. So they sent out an EpiX saying, hey, we think we've identified clusters of CRPA. HAI, AR programs need to be on the lookout for if you're hearing about CRPA outbreaks in healthcare facilities, and then also saying to the labs, you need to reach out to your partners in the clinical labs, and if they have carbapenem-resistant Pseudomonas aeruginosa, they need to be submitting those isolates to the public health lab. So the EpiX was really a way to kind of like mobilize, let everybody know in every state, hey, we've got something going on, and making sure that people are vigilant and looking out for it. Because otherwise someone might just be like, oh, CRPA, not thinking anything of it. And we see CRPA VIM all the time, but just not VIM80. We see other variants of VIM. So the EpiX was sent out in October. And in between October and the end of the year, CDC were obviously continuing to go through this extensive Epi investigation, which I'll talk a little bit more about, to identify the common exposure. And when they were thinking it was tears, they also notified ophthalmology societies so that they could be communicating to their healthcare providers about this. And it wasn't until February that the FDA actually issued a warning to consumers and healthcare providers not to purchase and stop using any EzraCare artificial tears or Delson Pharma's artificial tears that they had. I'm going to mute that person. Okay. was very distracting. Okay. All right. So based on the fact that we knew that this outbreak was going on, what actions were taken? And really, this is just to highlight that lab testing that took place in the public health lab really led to actions that others were able to take. So CDC worked with states on doing retrospective and prospective case finding. As I already mentioned, encouraging the submission of CRPA isolates to public health labs and the Antimicrobial Resistance AR lab network, which I'm going to talk a little bit more about later. The CDC then also did an extensive EPI investigation, which involved chart review of current patients to determine products that they'd use. What was challenging about this outbreak is that there were multiple outbreaks with unrelated geography or proximity. And so it was indicating a common product, but then trying to figure out what that common product was challenging. As I said, 2 of these clusters were in long-term care facilities. One of the other larger clusters was in an eye clinic. And it was like, why would it be an eye clinic and then long-term care facilities? Some of the specimens, the isolates, some of the specimens were eye sources and others weren't. So it was kind of confusing to try and figure out what was going on. But the other thing they did was doing screening of contacts at these locations where clusters had occurred. So part of what the Antimicrobial Resistance Lab Network does is when it identifies antimicrobial resistant organisms in a healthcare facility, it might go in and say, for example, screen everybody on a floor. So they went in and did some additional screening, looking for cases. This is a map from that CDC presented in March of 2023. So this was data from, that they say from May 2022, because like I said, they went back and then did more retrospective case findings. So this then went back to May 2022 up until March 2023. And there were 68 cases, and 37 of these were part of 4 clusters at 4 facilities. And as I said, specimen sources really varied. Some of them were surveillance specimens. So that's where they went in and did screening. So it could be like axillogroinal rectal swabs that just came in for screening for antimicrobial resistant organisms, as opposed to someone who had some kind of fulminant infection that they were trying to figure out what it was. So that was 46%. 26% were from eye sources. And then 35% were other sources, which had a whole range as well: urine, sputum, blood, wound or tissue, and body fluids. So it was really a mixed bag. And you can see all of the states involved in this outbreak, Florida included. And this was just up until May of 2023. And I'll give you a more current update in a minute. So as I said, the CDC went out and did this case control study at the Connecticut long-term care facility. And so they were looking at all the products that the patients used, all the contacts that they had, just trying to figure out what was going on with this. And they figured, okay, it's got to be a common product because we've got LA County Eye Clinic, long-term care facility, Connecticut. And what they were eventually able to do, and I don't know what magic they did to figure this out, but they realized that patients had 5-fold odds of exposure to artificial tears. So that was cases of patients that had CRPA. So then they went to all of those patients and were like, okay, did you use artificial tears? Most of them were saying yes. What was interesting was there was also things like patients that were diabetic, for example, and were using, it wasn't because they had an eye infection or anything like that, but they were using artificial tears because diabetics quite often have issues with their eyes and needed to use artificial tears. So I think just figuring out how many people actually use artificial tears for things other than eye issues. But the other thing that was happening was patients would bring in their own artificial tears, as well as potentially having artificial tears provided to them by the healthcare facility. And most people couldn't remember. They were like, yeah, I used artificial tears, but they couldn't remember the brand. So then they're like, oh my gosh, we have like 40 brands out there and no one can remember what they use. And they were like going back to people saying, look at your Amazon purchasing history. And they'd be like, oh yeah, go back to that and you know, figure it out. So, but I mean, amazing disease detectives. That was really disease detectives at CDC doing all of this stuff and figuring it out, and found out that the largest manufacturer of tears related to these patients was Ezricare artificial tears. So what are artificial tears? Well, in this case, they are meant to be, so instead of it being like a preserved product, they're preservative free. They can come in single-use vials, but a lot of these Ezricare artificial tears were multi-dose. So you open it, you use it, it has a dropper, you could close it, use it again, which obviously is gives the potential for it to get contaminated. And the Ezricare, the product was manufactured in India and was sold throughout the US. And a very common, and obviously you can buy it on Amazon as well as being able to get it, you know, from healthcare facilities. So as I said, by February of 2023, there was a recall from FDA. And so FDA sent out the consumer warning. And Global Pharma, that was the producer of these artificial tears, actually initiated a voluntary recall. So that recall covered all unexpired lots of, and also expired lots too, but I don't know why unexpired, but unexpired lots of Ezricare artificial tears and Delsum Pharma's artificial tears. So what's the status today? Well, as I said, after that recall, Global Pharma Healthcare actually expanded the recall and added Delsum artificial eye ointment cream. As of May 2023, there were a total of 81 patients in 18 states. Some of the additional patients were cases where patient specimens were collected before the recall. So some of that retrospective case finding where we knew we were going to find additional patients. But some were from after the recall. So obviously people had Ezricare artificial tears and were still using them after the recall. Some of these people resided in long-term care facilities, and they did report using artificial tear brands. Most of the patients had serious underlying conditions. Most of the eye patients had underlying eye conditions. 3 patients died, 8 patients had vision loss, and 4 patients had enucleation, which essentially is the removal of their eyeball. So pretty serious impact from this outbreak. CDC performed testing on opened bottles of Ezricare tears and found 23 bottles tested, 11 had bacterial growth, 7 matching the outbreak strain. FDA, it's funny the way things get done here, but like if it's unopened product, or let's say when we've had outbreaks before with things like tattoo ink and things like that, if it's opened ink, CDC does the testing. If it's closed and you're getting it from the manufacturer, FDA does it. So so CDC did the testing on open bottles that came from those healthcare facilities, or either from a patient from their home, like, hey, do you have any of that left over? CDC did that testing, but FDA did all the testing on the unopened bottles. FDA also did an inspection of the production plant in India and found contamination issues and several safety lapses, which they said, okay, you know, in order to continue to sell, you've got to fix X, Y, and Z. But you know that this investigation is still ongoing. It hasn't ended. I know that I talked to the lab in New York a couple of weeks and they were like, yeah, we just had another case. And obviously it takes a couple of weeks sometimes for, you know, by the time you've got the isolate from the patient and then you've done all of the appropriate testing, it's got to navigate its way from like a long-term care facility to potentially a clinical lab, then to the public health lab. So the other thing is, because these types of organisms, and we're going to talk about that, can be easily spread in healthcare facilities. So yes, somebody may have used artificial tears, but if they then get an infection or sepsis or something with one of these organisms and they're sharing a room with somebody else in one of these long-term care facilities, it's very easy to spread it to other residents of these long-term care facilities, especially when we think about healthcare associated infections, you're thinking about vulnerable hosts, and these long-term care facilities have a lot of vulnerable hosts because these people have underlying conditions. So how do labs detect antimicrobial resistant organisms, and what actionable results does this testing provide? So I mean, that whole example of that outbreak I gave you is just to come around to saying, well, how does the public health lab and the antimicrobial resistance lab network play a role in detecting antimicrobial resistance, providing actionable results that ultimately limit and prevent outbreaks, particularly in healthcare settings? And you can imagine with this tears, these artificial tears, that these infections could have been just ongoing for so much longer. And one of the questions we were all like was, but what if it wasn't a resistant organism? It could still have been a bad bug and in artificial tears, but we wouldn't even know it was ongoing, because the reason we picked this up is because we were looking for antimicrobial resistance, and this had it, and it had a unique gene. But what is that to stop contamination of multiple products that we use? So obviously the focus here is antimicrobial resistance, but it does get me thinking about the widespread use of certain products and infections that they can cause. So I think it's good to be vigilant, and I think, you know, we all do that in our labs anyway. Would be like, hey, I've noticed an uptick in such and such organism, or hey, you know, and that's often through our networking and our communication together is how we figure out that something might be going on and there might be an outbreak. But let's focus on the Antimicrobial Resistance Lab Network. So this is a CDC initiative, and it's a coordinated approach to combating antimicrobial resistance, impacting infection control, prevention, and antimicrobial stewardship. And the diagram on the right is kind of to show you that this is meant to be really a back and forth coordinated approach between multiple entities, healthcare facilities, labs, both public health and clinical, and of course commercial labs, health departments, CDC, all working together in a coordinated fashion to detect and prevent AR. And this is a schematic from the AR Lab Network. All of this information, if you're interested and you don't know about the AR Lab Network and want to learn more, just go onto the CDC website. It has a whole AR Lab Network page with loads of information, loads of resources. It has a recap on all the different outbreaks that the AR Lab Network has worked on. But as you can see, the AR Lab Network is divided up by region, and in each region there's a regional AR lab, and for us here in the Southeast, our AR regional lab is Tennessee, and then for the mountain region, Utah, central Minnesota, west is Washington, Midwest Wisconsin, northeast is Wadsworth Center, mid-Atlantic Maryland. And so that's it. It was an interesting experience for me to go to Wadsworth Center for 2 years and be in a regional AR lab and learn all about the stuff that the AR labs do. They do a ton of sequencing, which is really exciting. And for the other states that are in that region, you still do testing for the AR Lab Network, but then there's a lot of stuff you then refer to your regional lab. So actually, when I was saying about things taking a while to get to the lab, I guess, you know, it's from a healthcare facility to maybe a clinical lab, to their public health lab in their state, and then maybe to a regional lab in their region. So, you know, for example, Wadsworth Center did all of the testing for that Connecticut long-term care facility because they're the regional lab for Connecticut. But even, you know, in individual states within those regions do have some capability to do testing for AR. But if there's other things beyond their capability, they're then able to refer to the region. And the other important thing that came out was this publication in 2019, which was CDC AR threats report. Again, if you haven't seen this threats report, it's a very interesting read, but we'll tell you about all the significant AR threats that we should be concentrating on as laboratorians, as infection preventionists, as pharmacists prescribing drugs. So the way that the threats report is designed is organisms are listed as urgent threats, serious threats, concerning threats, and then at the end there's like a watch list. So what's on the urgent threats list? Carbapenem-resistant Acinetobacter, so we think crab. Candida auris, we know all about Candida auris in Florida, big problem. Clostridioides difficile, carbapenem-resistant Enterobacterales, and drug-resistant Neisseria gonorrhea. But two of the things, well, I'm going to focus a little bit more today on carbapenem resistance, so talking about crab and CRE, and then under our serious threats, obviously talking about CRPA, which in 2019 was a serious threat, multi-drug resistant Pseudomonas aeruginosa. I wouldn't be surprised if CRPA moves up to urgent threat for the next threat report that comes out, whenever that does in the next couple of years. But a whole list of other things on these serious threats: drug-resistant CAMPI, ESBL producers, VRE, drug-resistant Salmonella, Shigella, MRSA, drug-resistant Strep pneumo, and drug-resistant tuberculosis. And then in terms of concerning threats, Group A strep and Group B strep resistant organisms. And on the watch list is azole-resistant Aspergillus fumigatus, drug-resistant Mycoplasma genitalium, and drug-resistant Bordetella pertussis. So, I mean, I think for us anyway in the lab, we'd probably just put them all on the urgent list, because these are all things that we certainly are concerned about every day in the work that we do. But as I said, I'm going to talk a little bit more today about carbapenemase resistance. This is one of the main focuses of the AR lab network. And really looking at detecting carbapenemase producing organisms. So that, as I said, is crab, CRE, and CRPA. And I wanted to just explain a little bit about the difference between carbapenemase production and carbapenem resistance. So carbapenemase producing organisms actually harbor enzymes that hydrolyze carbapenems and other beta lactams. They're often found on mobile genetic elements, so we're thinking plasmids, which means that they can be freely shared with other bacteria in the vicinity, and are commonly associated with healthcare-associated infections. Because of their mobility and their ability to spread, that's why we have a real concern about them in HAIs. However, bacteria can be resistant to carbapenems through other mechanisms not mediated by a carbapenemase enzyme. And these are carbapenem-resistant organisms. So we'll call those CROs. And I think it's very important to know the difference between CPOs and CROs. So a carbapenem-resistant organism that doesn't have a carbapenemase may mediate resistance through a mechanism such as porin loss that Lisa just talked to us about in her introduction about Fetroja, which I loved learning about that name, and efflux pumps. And when we look at the data of carbapenem resistant organisms that are coming into Wadsworth Center, only about 25% of those are actually carbapenemase producers. And they're the ones we care about, because those are the ones on plasmids jumping all around the place, causing these problematic HAIs. So one of the roles of the AR lab network is to distinguish between what's a CRO and what's a CPO, because we want to concentrate on the CPOs. And so often we'll get resistant organisms into the public health lab, and we are able to confirm whether something's a carbapenemase producer. So like I said, the AR Lab Network Public Health Labs, Bureau of Public Health Labs here in Jacksonville included, focus on these CPOs. The labs will then perform diagnostic confirmatory testing and other surveillance testing for these CPOs. And submission of samples can include isolates that are coming in, as I said, for confirmation of carbapenemase presence or production, or it might be primary clinical samples coming in to screen for carbapenemases. Like if you know that in a specific healthcare facility, you might have something going on, you might want to go in and do a screening. So here are the criteria for submission to the AR Lab Network. For CRE, it's resistance to imipenem, meropenem, or doropenem at an MIC of less than 4 micrograms per mil, or greater than 4 micrograms per mil, or greater than 2 for ertapenem. And for CRPA and CRAB, it's resistance to imipenem and meropenem or doropenem at MIC of greater than 8 micrograms per mil. For the criteria for sending in a primary clinical specimen instead of an isolate, which for carbapenem resistance is a rectal swab. This is, as I said, when it's screening for carbapenem resistant organisms in healthcare facilities, which is always in conjunction with an infection prevention team. So that's one of the important things about the AR Lab Network. And when I was showing you this whole coordinated approach, health department, CDC can't just walk into healthcare facilities. That has to be a coordinated, mutually agreed upon process. And, you know, those healthcare facilities essentially have to invite the health department in if they want assistance with the screening. So really working in conjunction with an infection prevention team and the health department, there's a missing T, to target close contacts of a known carrier. Or it might be to do a point prevalence survey. You want to know what is happening on my unit at this moment in time. Or some facilities know they've had a problem in the past and want to do either admission screening. They want to know what their patient has before they come in, or it might be discharge screening, because what happens a lot with these patients is they're going between multiple healthcare facilities all the time. They could be going from a hospital to a long-term acute care hospital, or then to a long-term care facility. So admission and discharge screening can be really important in this situation, especially if that facility knows that they've had an issue with a certain organism. So just to run through the testing algorithm at the public health lab, if an isolate comes in, MALDI-TOF is performed for ID, and then phenotypic testing can be performed to confirm if it's a carbapenemase producing organism. There are a couple of methods that are commonly used out there, either the MSIM, which is the modified carbapenemase inactivation method, or the CARBA-NP. And then you can also do some molecular testing to look for carbapenemase production. There's the Cepheid's Expert CARBA-R and the NG test CARBA-5. And then in terms of surveillance testing, so if a primary sample comes in like a rectal swab because you're doing screening in a unit at a healthcare facility, that again is usually a molecular test, either the Cepheid Expert CARBA-R, or some public health labs do their own lab developed test to do this surveillance screening. And then antimicrobial susceptibility testing is a whole host of things, disk diffusion, gradient diffusion strips, but all of the regional AR Lab Network labs also do broth microdilution panels with Sensititer. And the regional labs have expanded AST panels where they can do additional drugs that might not be available in the clinical sector. So if you have a really resistant organism and looking for expanded AST panels, the public health lab can do that as well. And at Wadsworth Centre, which was a regional lab, they also had a 3D printer. They could make their own broth microdilution panels and add the drugs they wanted to it, which was pretty cool. So just to focus on the molecular assays and what they're actually looking for. So we always think of carbapenemases as there being like the big five. So there are KPCs, which are class A serine beta lactamases. There's NDM, VIM, IMP, which are class B metallo-beta lactamases. And then there's OXA48 like, which are class D oxacillinases. So those are the 5 main carbapenemases that we're looking for with carbapenemase producing organisms. But then you might want to do some additional molecular testing. For example, if you're looking at carbapenem resistant Acinetobacter baumannii, you also want to be looking for OXA23, OXA2440 and OXA58, because these are more commonly seen in crab. And these are also oxacillinases like the OXA48 above. Now, whole genome sequencing is extra awesome because it will detect all of the carbapenemase genes and also provide additional analyses. Like I said, it can provide you with a sequence type. It's not just going to tell you it's a VIM, it's going to tell you which variant of VIM it is. So like we were saying with the CRPA, that it was VIM80. And it can even tell you things, it'll give you an indication of where that gene is located, whether it's on plasmid or chromosome. So why do we love whole genome sequencing? I mean, I think Jam has probably had lots of whole genome sequencing talks, so you're probably already converts. But anyway, enhances detection of emerging and novel AR threats. As I said, provides sequence type, gene variant, plasmid type, and maybe sometimes can indicate other virulence factors that are at play. It can provide surveillance data on geography of circulating strains in your jurisdiction. It can determine relatedness between 2 or more isolates. And it really provides high discriminatory power for determining transmission events and outbreaks that impact infection control practice and public health action, and can be used in conjunction with that epi data to really build a whole picture. So this is an example of a whole genome sequencing pipeline, really just to give you the just simple overview, a step-by-step process. This is just the pipeline from Wadsworth. They do mostly Illumina sequencing there and some MinION Oxford Nanopore sequencing. But for the AR stuff, it's Illumina sequencing. So they get the sequencing reads, trim the reads for quality, then run it through the pipeline where it will identify the species, which you already know because you had your isolate and you ID'd it by Moldy, you know what it is. And that just lets you know, okay, my pipeline's working, I've got the right species ID. And then it'll do assembly of those sequencing reads in QC. And then it will do an MLST typing that, like I said, you'll get your sequence type. And then it will look for antimicrobial resistance genes. And the pipeline at Wadsworth used 3 freely available databases, ResFinder, CARD, and the NCBI AR database. And lastly, the pipeline will also identify whether there is plasmid there and other virulence factors. So as I said, these databases that can be plugged into your AR pipeline are all freely available. CARD, ResFinder, and the NCBI AMR Finder. And like I said, of course, your first step is to do the sequencing, and your pipeline is going to verify your ID, give you sequence type, identify gene variants. And then there's kind of like 2 pieces with the AR pipeline. One is like, let me just look at this as organism, at this assembly, ID it and say what's there. The other piece is then, okay, let me compare it to everything else I have in my database. So then it's going to be looking at anything else in the database that's similar. It can look at mutation single mutation events or single nucleotide polymorphisms, or it can look at nucleotide insertions and deletions, called indels, and then it's going to identify genetic differences between isolates that are in your database. The other thing I should say is, as I said before, most of this is done with Illumina sequencing, but you can, which is more short read sequencing, which is harder for looking at things like plasmids. So you can do additional sequencing, long read sequencing, if you prefer, with something like MinION from Oxford Nanopore Technologies, and that will help you do additional analyses on the exact location of the AR gene, plasmid versus chromosome. So just to look at kind of thinking about and visualizing relatedness when we're looking at that side of the pipeline, you can kind of visualize it in different ways. You can either do like a matrix that has the number of mutation events, or SNPs. So on the left-hand side, you see that SNP mutation event matrix, and the number is literally the number of single nucleotide polymorphisms. So that's mutation events different between two organisms. And each line on that matrix represents an organism. So obviously, this organism compared to itself is 0 SNPs, but this organism compared to this organism is 2 SNPs. So essentially what you can do is line them up and see which ones are closely related to others. This can also be visualized in a heat map, which is basically exactly the same thing as the SNP matrix, but it's color-coded. So the number of SNPs difference, it starts to get lighter, the less related they are. And then you can see on the diagram on the right-hand side, you can also visualize this in kind of a phylogenetic tree graphic as well, if you want to see how they're related. But I think one of the things that's challenging about doing all this sequencing and in the AR lab network and working with the HAI and AR programs is often working with EPIs or HAI programs and infection preventionists. And you can give them all this data and tell them about sequencing and SNP matrices and all this stuff, but they're just going to be like, tell me what it means. Like, are they related to each other or not? So I think one of those things about communicating from the public health lab to those healthcare practitioners so that they can understand the data that we're trying to present to them. So lastly, I'm going to finish up just giving you 3 examples of how whole genome sequencing has supported detection and prevention of antimicrobial resistance through the AR lab network. We talked already about this CRPA sequence type 1203 Vim80 GES outbreak. Another example is identification of a novel carbapenemase gene. And lastly, look at relatedness analysis of isolates in a healthcare facility. And that last one as well is going to come back to how do we try to visualize to help this make sense in a healthcare facility to infection prevention so that they can take action. So just in summary of the CRPA outbreak associated with artificial tears, I mean, this, of course, whole genome sequencing was the way that we knew, hey, hang on a minute, this is an unusual carbapenemase previously not seen in the US. 5 years ago, we would have just said it's CRPA Vim, and we would have said every Vim was a Vim. There wouldn't have been any of the, oh, it's a Vim80, it's a Vim20, it's whatever. So having whole genome sequencing was enabled us to say, this is Vim80, we've never seen it in the US, what's going on? Whole genome sequencing can give you the sequence type at the same time, which at least is a high level. Well, are they all of the same sequence type? Before you even get into the genes, you're like, okay, are these possibly related? Because the other thing is these organisms, some of these genes do come and go. And with this particular case, obviously whole genome sequencing was able to match the clusters in LA, Utah, and Connecticut at the beginning of the outbreak. I mean, I think if we think that cases started in May, and by September, they knew that these clusters on opposite sides of the country were related to one another is pretty impressive, and ultimately helped them to be able to find a common source. Additional cases based on submission of specimens from screening efforts and of CRP isolates from healthcare facilities. Once this outbreak had been identified, it was then much easier to go out and find retrospective cases and make sure people were on alert to find prospective cases. Okay, what about identification of novel carbophenamases? Well, this is a case from Wadsworth Center. So they had a Pseudomonas aeruginosa identified by MALDI, and it was MSIM positive, meaning, okay, this is a carbophenamase producing a Pseudomonas aeruginosa. But when they did the molecular test for the top 5 carbophenamases, it was negative. So that would be KPC, VIM, IMP, NDM, and OXA48. So the PCR was negative. So then like, okay, well, we know it's a carbophenamase producer, but it's not the top 5. So they did phenotypic susceptibility testing. They were like, this is definitely a carbapenem-resistant organism. But without sequencing, we wouldn't have been able to figure out that this was actually a novel carbapenemase. And it was the first BLASIM isolate that was identified in the United States. And we may think, okay, why is this important? Well, we want to have an understanding from doing our surveillance of what's going on, where things are spreading in the country or in your jurisdiction, and obviously, ultimately, how we can stop it. But it's obviously very important to keep being able to detect and stop these novel carbapenemases that surely will, I'm sure, keep popping up. Okay, so lastly, identification of transmission in a healthcare facility. So what was interesting in this case is this is in a healthcare facility, 1 unit on a floor. What was interesting here, and what I was going to tell you about, is you might have a ton of plebpneumos on one unit and think, oh, well, I have 5 patients with plebpneumo, they've got to all be related. And whole genome sequencing can really help you figure out what's going on in these situations with much more high discriminatory power than what we used to have. I mean, I think even, when we think about when we used to do PFGE for nosocomial infections, even though PFGE was great, Matthew, wasn't it? But but whole genome sequencing just gives you so much more accurate information. So in this actual case here, there was an index case that had plebpneumo, and it was actually an OXA48, which is a bit unusual. But then there were 3 patients that had KPCs that matched, and then there were another 4 patients that were also KPC, but were not the same KPC. So the Ys were all related to one another, and the X's were all related to one another. And then the ones that don't have anything were KPCs, but weren't related to anything. So you can see a ton of the green dots that represent KPCs. Only a handful of them are related, and that really helps infection prevention figure out what's going on. Otherwise, they could have thought everybody there had KPC, they're all related, I've got to move these people, I've got to isolate these people, this person needs to be on contact precautions. I mean, obviously they had a bit of a KPC problem, but they weren't all spreading it to one another. Obviously 2 of the X's shared a room. One of the X's was in a room next door. But when you are doing infection prevention and trying to figure out, are these patients spreading it to each other, or is this a healthcare professional spreading it from room to room? This is the kind of information that you need. So whole genome sequencing can really enhance our ability to detect and perform surveillance for AR in the US. As I said, it provides higher discriminatory power than previous methods. and relatedness analyses for infection control and prevention purposes. I guess I should also say here that these kind of visuals are much easier for infection prevention, but often we don't get to this point with this visual for like a little while after, because deciphering all this information and doing the diagram and doing all of this stuff with our epi team takes some time. But it's a nice way to visualize things, but often after the fact. But yeah, provides relatedness analyses for infection control and prevention purposes. And it provides temporal and spatial analyses. So we think about where are we seeing these and when are we seeing them, and really like putting that together. I mean, if we go back again to the CRPA outbreak, if we just looked at geography, we might not have been able to piece these things together because geographically it didn't seem to make sense. But obviously, temporally it did, because we suddenly had all these CRPAs at the same time. So I think looking at all of those things and doing surveillance can really help in determining outbreaks. And I think that the PulseNet program, which you probably heard a lot about, I'm sure Matthew's talked about it, Kelly, but we can learn a lot from the PulseNet program, which is a national effort looking at food and waterborne bacteria, and enables you to detect multi-state outbreaks for those. I think the AR Lab Network's really been able to build on this long-established PulseNet network, which I think started 1996. Does that sound right? It's 90. Anyway, it's pretty old. So and that all used to be with PFGE, and now it's all with whole genome sequencing. So, I think the AR Lab Network is going to be another of these really great national surveillance platforms that we're really going to rely on to do a lot of our AR diagnostics and surveillances. And obviously whole genome sequencing can detect and track novel strains. And if we have anything, on our high concern list, or our EPIs reach out to us and say, hey, watch out for this, we can look for it with our whole genome sequencing analyses. Public health, this is a very cheesy line. Public health labs perform essential testing, but just to say about how yes, we have our regional labs doing some more specialized diagnostic and surveillance testing, and doing a lot more of that tracking across their whole region, but the state labs that are part of the AR Lab Network are also providing a really essential role doing diagnosis and surveillance, and that's happening under Kelly at the Bureau of Public Health Labs in Jacksonville. And as well as doing the testing, there's also a lot of referral that's going on. Or if like someone comes in and they go, we want to do screening for Candida auris at such and such facility, then we here in the state of Florida have to coordinate getting all of that testing, because it actually gets done at our regional lab in Tennessee. So it's this huge national coordinated approach that enables public health to detect and respond to public health threats more rapidly, and can cohesively work with healthcare, infection prevention, and health department HAI AR programs. So that's it. Obviously acknowledge the Bureau of Public Health Labs, Colleen and Suzanne, who helped with these slides. And I should say thank you to Kelly, because it's in her shop. And then Wadsworth Center, New York State Department of Health, Kim Musser oversees all of those labs, and Liz Nazarian's the person that leads the AR regional lab for Wadsworth. And then some of this data came from CDC, from their ARX, which is the antimicrobial resistance, and then their DHQP, which is one of their quality teams. And Maroya Walters at CDC was one of the main people involved in this investigation, and some of the pictures are from her. So that's it, and that's my contact information. If any of you ever need to, it doesn't have to be about this, but need to contact me for anything, there's my email, and there's my cell. So, and I'm happy to have any questions if you have them.
So you talked about the transition and you said that obviously building the map, they couldn't really build the map until after the fact.
So you're talking about transmission and you can't really build the map of where everything goes until after the fact. Where are the bottlenecks in this process
Of building the maps.
of being able to do that sort of thing essentially in real time?
Well, we can do things like the SNP matrices, and we do those in real time, and we can communicate those to our HAI-AR programs. So those are like Department of Health epidemiologists, and usually it's kind of their role to communicate with infection prevention and explain to them. And, you know, sometimes I don't know what we do here in Florida or what Tennessee as a regional lab does, but in Wadsworth, there'll also be text that explains what's going on, where it'll say, these are considered related, and it will give you the accession number and the isolate. So you can see all of that getting reported. What I think there are two bottlenecks. One, often collecting all of this data and piecing it together happens afterwards, right? Because at the beginning of it, you've maybe got 1-2 KPCs, you're still trying to figure it out. By the time you've done your screening and figured out, hey, I've got 10 KPCs on my unit and they're related, you're weeks down the line. And this is also happening, you know, you're thinking about infection prevention going in and doing testing, infection prevention going in and doing screening. So, you know, Wadsworth was also involved in a huge MSSA outbreak at an academic medical center in New York City in a PICU, and it kind of just took weeks with collecting specimens and figuring out who was in which patient was in which room and what date was the sample collected. Was it a screening test? Was it an isolate that was sent in? Just piecing that information together, I think, just takes time, and everyone's also doing everything else in their job as well as this. But in more real time, yes, that is getting that information from the lab is getting communicated to health department HAI-AR programs about, hey, we've got these isolates, we think they're related. The second bottleneck is most public health labs can't clinically report relatedness data. Like, that's not a clinically validated test. So the way that CMS views it is that if you take any patient action, then it needs to be a clinically validated test. So we could argue about what that action has to be. To me, it would be like prescribing a drug, but I think there is some argument that if you then said, oh, hey, you said these two were related, and then you moved a patient to a different room, they might consider that as being a patient action. So I think all of us are struggling right now with, what do we need to clinically validate with whole genome sequencing, and what can we put on a report that goes out through our LIMS versus what we're doing right now, which is communicating to our EPIs and saying, hey, this is what's going on, and then they kind of get with infection prevention. So those are the bottlenecks.
Questions on the chat? No.
No, I think it was just about, could you hear?
What was the most interesting project you were involved with in New York?
The CRPA, this outbreak was pretty cool, because I literally remember the day that Liz Nazarian came to me and said that she had identified a VIM80GS9 and it had never been seen in the US. We were like, tell CDC, tell CDC. But they didn't, they were like, oh, for like about a week, I'm not lying, because it was just a one-off, and it's just, you know, then when it was like, hang on a minute. And the other thing that was exciting about that outbreak was we just couldn't figure it out, because we were like, why is this one eye, this one's sputum, this one's a wound, this was a rectal swab in someone that was asymptomatic. We just like couldn't figure. And then the LA County one, they were all in an eye clinic. So you're thinking, well, it has to be something to do with the eyes. But then all the others were not necessarily related to the eye. So I remember before they figured out what the product was, we were all just scratching our heads like, I don't know how CDC figured it out, but some very smart people there.
Thank you. Nice to have you back, Mary Claire.
Huh?
Nice to
Oh,
have you
it's good
back
to see
in
you, Therese.
Jacksonville, although I've not been active myself. With regards to the FDA inspection of the laboratory, the pharmaceutical lab in India, were they invited to come there and inspect? Did they go there and inspect because they thought they had jurisdiction since this eye drop was being brought into the
Well, I
United
think
States?
I think they have jurisdiction if it's being sold in this country.
That's what I said. That's the only reason they would be able to go there and inspect this lab.
Yeah.
Because it was being sold in the United States.
Yeah.
Second part of that question is, it said that your presentation said that they found some contamination issues. Did they find this actual Pseudomonas organism there, or other organisms as well? Or do
So
you know?
I haven't seen anything officially reported, even though they did find, from what I understand, they did find bacteria. I don't know whether they ever confirmed that it was CRPA. Sometimes FDA is very tight-lipped about this. I imagine there will be a full report, published report that will come out on this, but even CDC hasn't even issued an MMWR publication on this yet, possibly because it's still kind of ongoing. So I never even saw data from the unopened artificial tears. All I know is that they did find bacteria, but I don't know if they found CRPA. Was it FIM8TGS9? I don't know.
Did find anything there, or how are they related to the lab that
The second manufacturer?
manufacturing?
It's one manufacturer just sold under two things. So it
Okay.
was Esricare and whatever the other one, Delsum.
Sort of
But Global Pharma manufactured both of those. Yeah. But if you Google it in the news, I think it talks a little bit more about their inspection in India. Don't use Esricare or Delsum.
Well, Maria Claire's not leaving us anytime soon. She'll be around for questions for a while,
Yeah.
I hope.
I will. Yeah.
Any last ones?
No, I don't think so. Thank you all online.